Hi everybody, I am RAMUKATTUroju. This is our channel NEET Botany in 20 minutes. Today we have to discuss about transforming principle. It was given by Griffith, so it is also called a Griffith transforming principle. This Griffith transforming principle, it is very important principle. It was using in genetics and molecular biology. Here it was given by Frederick Griffith. In 1928, Frederick Griffith was did the experiment on Streptococcus pneumoniae. The bacteria here, Streptococcus pneumoniae. It was caused pneumonia disease that leads to the death of the organisms. First, Griffith was grown this Streptococcus pneumonia in a laboratory in petri dishes. He has observed two strains of Streptococcus pneumonia. One strain is having the wall. The wall will be ridges and furrows, means it is in a rough texture. It was calling as a R or a rough strain or a R strain. Next, another one is having the extramucilase coat around this bacteria. Extramucilase coat will be formed. This strain is calling as a smooth strain because it appears smoothly. From externally it appears smooth, so it is called a smooth strain. This smooth strain is also called a S strain. This S strain having a mucilaginous coat. This coat is made by the mucilase. Mucilase coat. By the mucilase coat, the S strain appears smoothly. But actually, smooth strains are having extra protection than the rough strains. First, the smooth strain was applied into the mice, means injected into mice. It is the first experiment. Here S strain injected into mice. Injected into mice. When injected into mice, mice was died. Because when strain is entered into mice producing the digesting enzymes, but strain has the extramucilase coat, so it can protected by the enzymes and it attack on the organs of the mice, and finally mice will be died. From these mice, they are also collected the S strains. S strains are collected. Okay. In next experiment, he was taken this R strain, injected into mice. R strain injected into mice. Now mice was live, and also they did not get any strain. No strain was present in the body of the mice. Now they are killed this S strain by the boiling water, by the heating of S strain. It was killed by the boiling water. Killed S strain by boiling water. Means by the heating it was killed. So now in third experiment they are taken this heat kill assistant injected into mice. Heat killed assistant injected into mice. Now mice was live and also not get any strain from the mice body because it was already killed, so it was not fight with the enzymes. So enzymes are digested the assistants, means heat kill assistants. Now in a fourth experiment, what they do means here if R stain injected into mice live, heat kill stain injected into mice live. So these two are mixed and injected into mice. These two are individually will not affect to the mice, but these two mixed and injected into mice, what will happen? You see here, heat killed S strain and R strain, normal R strain. What will happen when injected into mice? Injected into mice. Now mice was died. Mice was died. And also, shockingly, he got the assistant, living assistant in a mice body. Actually, living assistant is not injected, but living assistant was present. How it was formed means by this Griffith was said that here some material is transformed from the heat kill assistant to the R strain. Some important material was transformed. When important material is transformed into R strain, the R strain becomes into S strain, means forming of extra coat. R strain becomes into S strain. Now the S strain is fight with the mice enzymes and not affected. That means enzymes are not affected on S strain. So S strain is killed the mice. Means in this experiment, what he want to tell means here, some material, important material is transformed. That is maybe genetic material. Okay, that was given by the Frederick Griffith by this transforming principle. Later, by this experiment, the three scientists like Avery, McLeod, and McCarthy, these three scientists are continued this experiment. Means here, this assistant which are came in died mice, they are tested, means examined by the applying of different enzymes like here proteases, RNases, DNases. When they are applied the RNases, the transforming is continued. When they apply the proteases, transforming is continuous. Means when they are applying proteases means protein digesting enzymes, means protein are completely digested. That part, remaining part entered into the injected into mice, the transforming is occurring. And the same here, the RNA is, when applies the RNA is also, here the transforming is continued. When applied the DNA is transforming is stopped. So by the above experiment, here Avery, McLeod and McCarthy said that DNA is the genetic material.